In this video, we will discuss about the chromatography and electrophoresis. As I have told that various types of techniques used in cell biology, for example, microscopy, tissue culture, chromatography, and electrophoresis. So, chromatography and electrophoresis in this video we will discuss. So, the chromatography word is derived from two Greek words, chroma mean color and graphy mean to write. So, the literal meaning of chromatography is color writing. And this technique is invented by a Russian botanist M.S. Sweet in 1903 while he studied the plant life for color material. And chromatography can be defined. It is techniques used in cell biology for separation of one type of molecule from other. Chromatography is a great physical method for observing mixture and solvent. In chromatography, basically composed of two phases. One is known as mobile phase, as the name indicates that they move with the sample, and it's basically composed of liquid or gas. In stationary phase, it does not move with the samples, and they have cellulose and silica. There are various types of chromatography techniques can be divided into various types. The first one is paper chromatography. In paper as well as thin layer chromatography, the mobile phase is the solvent. The stationary phase is the paper chromatography is the strip or a piece of paper that is placed in the solvent. Separation of chemical here depends upon the physical and chemical properties like solubility and molecular mass. In this picture, you see the apparatus we use in paper chromatography. This is various types of graduated cylinder having cork, and this is a paper. So this is basically the stationary phase, while these are the liquid in which various types of mixtures are present. So, and paper chromatography is used for the separation of photosynthetic pigment, sugar, and amino acid. In this diagram, you can see that various types of photosynthetic pigment, chlorophyll A, chlorophyll B, xanthophyll, and carotene, when they are present in the plants, and when we produce a solvent from it, a solution from it, a mixture is placed within the graduated cylinder, and with the help of a paper, the various types of photosynthetic pigment they move within the papers. As you see that the carotene they reached here and xanthophyll is present in there, chlorophyll A and chlorophyll B. So various types of photosynthetic pigment in the form of chlorophyll A, B, xanthophyll and carotene and sugar and amino acid can be easily separated by using the paper chromatography. The separation of hair chemical depends upon the physical and chemical properties like solubility and molecular mass in both paper chromatography as well as in thin layer chromatography. The second one is the column chromatography. The column chromatography is a method used to isolate a single chemical compound from a mixture. It is more commonly used method which involves the mobile phase flowing over a supporting matrix held in glass or metal tube, and it basically depends upon the differential absorption of compound and the absorbent. In this diagram, you see that column chromatography is one of the most useful methods for the separation and purification of both solid and liquid. This is a solid liquid technique. It is a solid liquid techniques in which the stationary phase is a solid, and mobile phase is a liquid. And this is used for small as well as for large scale. So this is about the column chromatography. These are various types of other chromatography techniques: liquid chromatography, gas chromatography, thin layer chromatography, paper chromatography, as well as column chromatography, as we have to discuss. The second techniques which are used in the cell biology or cytology is electrophoresis. Electro mean electricity and phoresis mean being carried out. It is also a type of biological technique for the separation of charged molecule under the influence of electrical field. Is known as electrophoresis, or we can define electrophoresis as a laboratory technique used to separate DNA, RNA, or a protein molecule based on their size and electrical charge. It have two types. Cataphoresis, as the name indicate, it is used for the electrophoresis of positive charged particle, and anaphoresis is the electrophoresis of negative charged particle. In electrophoresis, two factors are very important. Amount of charge. Greater the charge, faster will be the movement. Smaller the charge, slower will be the movement. And size of the molecule, smaller molecule they will move faster, while larger molecule they will move slow. So it depends upon these two factors in electrophoresis, amount of charge as well as size of the molecule. These are the apparatus used in gel electrophoresis. This is basically the electrode. This has buffer solution for the control of acid and base, and these are direction of movement, and these are sample wells, and these are basically the gel. That's why the electrophoresis in which the gels are used, they are known as a gel electrophoresis, and they are connected to the electricity. That's why this is known as gel electrophoresis. When we pour the materials, samples or mixture within the sample wells, when we apply electric field, the larger molecule they will move slow, while the smaller molecule they will move faster. So the smaller molecule they will be easily separated, and it depends upon the charge. If the charge are higher, larger, the movement of the material will be faster. You can see the result of that. If you like the video, please like, share and subscribe. Thank you.